Well, thank you very much. I'm thrilled to be here. It was one of my final pre-COVID activities was attending this event last year, and so it's really my pleasure to be here again. Oh, something just went wrong. Can everyone see that?
Yeah,
Great,
seems to be
thank
going
you.
okay.
Um,
Yeah, thank
so
you.
I want to add, hey, we are within 2 hours of the landing. We're at about 1 hour and 56 minutes. And I hope everyone here is as excited as I am. And I'm going to tell you one thing I'm excited about for this mission is the way that we can test the idea of that life on Mars, if it exists, could be related to us. And in doing so, potentially resolve some of the challenges of the history of life on Earth. So first, I want to start just by thinking about life as we know it, something that many of you are intimately familiar with. And there's three main components if we think about kind of the minimal aspects to life as we know it. One of those components would be metabolic. We need to produce, we need to harness energy, we need to do things in the world. Another would be informational. We need to pass on hereditary information in the form of DNA or RNA. And another one would be we need to form some kind of compartment to maintain the concentrations that we need to do all this, as well as to protect life from the world. And so one of the questions that comes up in origin of life research is what came first? Did we get metabolic aspects to life first and then informational, or was it the other way around? And so there's a whole bunch of different theories about the origin of life. And these are a few examples here. One would be that we know that many molecules relevant to life rain down from space in the form of meteorites and comets. And those primitive materials contain all kinds of the building blocks of life, including amino acids and ribose and other sugars. And so that might bias life toward using those kinds of molecules. Another way we can explore it is with artificial life. We can make protocells and try to figure out what is the minimal cell. We can also look at chemical systems that may have occurred in hot springs, in a surface environment perhaps, with UV light also playing a role in the synthesis of the first life. And then there's also this model of life originating at a hydrothermal vent. The most complete version of that, I would argue, is the alkaline vent hypothesis, but there are others as well. And so I'm going to focus in on one particular approach, one particular model of the origin of life, which is the cyanosulfidic origin of life. And the reason I'm going to focus on this is it does something really interesting, which is, and it's also plausible from a prebiotic chemistry standpoint. It does something really interesting. Cyanosulfidic chemistry, that is HCN, can form a reaction network in combination with ultraviolet light that produces the precursors to RNA, protein, and lipid. And because it can do this simultaneously, it can address the informational, compartment forming, and metabolic aspects of life at the same time. It solves this problem of which one came first. And so the hypothesis here is that there was this hydrogen sulfide, HCN and UV, which formed this reaction network and could have led to the first protocellular entities that could lead then to very much later our last universal common ancestor and the tree of life as we know it. Now, there's some additional challenges to this. It doesn't explain how we got to the point of fixing carbon, but there's some additional chemistry that could come into play. For example, carboxysulfidic chemistry. Now, I'm not a chemist, so there may be some, the chemists who are working on this are pretty bright and have come up with some really great models that seem plausible, and yet there's plenty of room for other models as well. But the surface origin is very appealing. It requires a shallow water environment and also wet-dry cycling. and it provides a mechanism that's very consistent with what we know about how polymers are made. In particular, polymer synthesis requires dehydration synthesis, and that's common to nucleic acids, lipids, and proteins. So we know from looking at sequence data and making comparisons of both amino acid and as well as DNA sequences that we are all life as we know it is related to a very ancient common ancestor. And we know that originally by looking at conserved DNA sequences, for example, within the RNA ribosome that translates RNA into protein. And when we look at that, it's like a stopwatch. There are regions that are highly conserved and there are regions that are less highly conserved. And by comparing those differences, we're able to build a tree of life. And in fact, of course, now we don't need to be limited to one gene to do this. We can look at, all conserved genes, and we can look at protein sequences, et cetera. And when we do that, we find that the archaeal root, the root of the archaea, might be older than 4 billion years. This can be done by comparing basically constraints in the tree of life with fossil evidence. Now, we have to take that with a grain of salt. There aren't a lot of fossils from long ago, and in particular, ones that we can provide definitive evidence of life. And so because of that, there are big error bars on this number. And yet, 4 billion years is a very long time, and it implies that this last universal common ancestor was even older. And by the way, the last universal common ancestor, we have a sense from the genomics, we have a sense of what that environment must have looked like. Perhaps geochemically active, rich in hydrogen, carbon dioxide, and iron. We also know LUCA had a complete genetic code. It was not something close to the origin of life. A lot happened between the origin of life and LUCA. Now, one problem is if we think about what we know about the early Earth was an ocean world. There was really no subaerial crust before 3.5 billion years ago. And in fact, it took, until 3 billion years ago just to have a couple percent. So this means that if there was a surface origin of life on Earth, it probably would have been limited to volcanic island hotspots. And so this raises the question, is that enough? Could life have evolved quickly enough in that setting? And if not, maybe we have to rethink our models, or maybe we have to think about an origin of life elsewhere. And this cyanosulfidic model, which is very plausible through all the different steps that need to happen, it really does require some time. We need deposition and accumulation of precursors and their transformation through geologic processes. So perhaps life could have begun on early Mars. So we can look at this and see that even if there was an ocean on Mars, then there was orders of magnitude more land area available. Now, another way that we can think about this problem is by looking at the genetic code and looking at what amino acids are present in meteorites versus in the genetic code. And if we do that, we see that the amino acids in the genetic code, especially the later ones, have different properties than meteoritic amino acids, as characterized here by the HOMO-LUMO gap, which is a measure of how easily electrons or how reactive a molecule is. And this decrease in the HOMO-LUMO gap means electron sharing is facilitated, and this corresponds with increasing complexity of the amino acids. Now, the argument that has been made here by Granold et al. Is that these amino acids are in order of the order that they were thought to have been fixed in the genetic code. The argument is the late ones may have been fixed in response to oxidative stress. Now, I told you before, LUCA was on the order of earlier than 4 billion years. But what we know about the history of oxygen on the Earth is that the Great Oxidation Event happened much, much later. And there was probably some oxygen earlier, but not really too much before 3 billion years ago. So there's two possibilities here. One is that there's a different reason that these amino acids were fixed. It's not related to oxidative stress. And an alternative would be that perhaps LUCA did not take place on the Earth. Maybe it took place in an environment that was more conducive to, that had more oxidative stress present. Okay, so what about an early Mars origin of life? We know today, based on prior knowledge from missions on Mars, that early Mars was likely habitable. It had all the elements required for life, variable redox states as identified in Gale Crater, probably more water than today, but not warm and wet, maybe still cold and arid. But actually, that could be good. This could enhance the accumulation of precursors and organic material at the origin of life. You might ask about other things that life needs. Where would that HCN come from? Lightning could have provided it. So nitrogen was available, some carbon was available, phosphate was also available. In fact, way more available on Mars than on early Earth. And Mars also had an earlier transition to oxidizing conditions that could potentially explain that fixation of amino acids in the genetic code. Sasolov et al. Have written a really nice paper that I would urge you to take a look at, The Origin of Life as a Planetary Phenomenon, and it walks through the processes that would be required for a cyanosulfidic origin and concludes that this origin could have taken place on Mars, on Earth, potentially, although they talk about the water world a little bit, and also on other worlds. We don't have any other good examples in our solar system that we can study today, so Mars is probably the best candidate that we have. Now, what would happen? Let's say there was an origin of life. What would happen then? Well, could life get from Mars to Earth? And there were a series of studies in the 90s and beyond that demonstrated the potential for the meteoritic transfer of life from Mars to Earth, so-called lithopanspermia. One of these was showing through paleomagnetism measurements that Mars meteorites could be ejected and end up on Earth without being heated. Indeed, ALH 84001 did not get above 40 degrees Celsius. Other studies, which involved putting microbes in rocks and blowing them up, that sounds really fun, demonstrated that microbes could survive the relevant shock pressures. And other studies were statistical in nature and concluded that viable non-sterilizing transfers on the order of 10 to the 12th kilograms had gone from Mars to Earth just in the last 3.5 billion years. And because their cratering rates have declined over time, we can expect that there would have been even higher rates of viable transfers earlier in the history of the solar system. Now, we also know that organisms that cluster near the base of the tree of life tend to be adapted to high temperatures. How would this be consistent with what we're proposing? Well, one possibility is that microbes that, say, hitched a ride from Mars to Earth experienced a selective pressure, and that selective pressure was being able to survive ejection from Mars. And that would naturally select for organisms that were adapted to at least moderate temperatures, mesophilic, and then in addition, survival on Earth during a time of many impact events could have also driven adaptation to warm temperatures. And this is some data from Grusin et al. Showing this molecular signal as estimated from protein sequences. So 45 degrees or confidence interval of 33 to 68 is basically consistent with ejection from Mars. Now let's get on. We're almost to our launch party time, so let's get cooking about Mars 2020. We're going to be going to Jezero crater, a 45-kilometer diameter crater, and with a lot of interesting deposits that are lacustrine in nature, that is lake environment. And I have to tell you, having been to an impact crater in the Arctic, this is a magical place, but then here even more so with all of these deposits. And here's why. The reason is on Earth, we don't really have a lot of rocks before 3.5 billion years, but here we can access rocks going back to even older than 4.1. Jezero is pulling in materials from this huge watershed of all these different diverse materials. So we can learn a lot about the history of Mars. So this is kind of putting together everything I've been telling you in the talk. Because of the current time, I'm going to suggest please go read my preprint on archive, and I would love to talk more about what makes sense here and what doesn't to you. And looking toward March 2020, what can we do? We can look for organics and high preservation potential materials. We can use these wonderful instruments, Sherlock and Pixel, to map out the presence and pattern of organic material, although we might be limited in unique molecular identification of organics. So to do that, what we can do with the March 2020 rover, we can look at the geologic setting of the source regions. We can see if the pH and oxidation are consistent with this model. We can look for certain kinds of organics. But really, to seek evidence of life as we know it, we will probably need future missions to target extant life in the subsurface. And for that, we are developing two instruments, the search for extraterrestrial genomes, targeting life as we know it, and another instrument, ELI, to target life as we know it or don't know it. And with that, I will say thank you to my lab, and I will conclude the talk. Thank you so much.